Hello guys, this is me Dr. Kashan Kasit on my channel EzyDoc. Today we are going to discuss the topic of the blood. Blood is a clear fluid. This is according to the normal people, but according to the doctors, we define the blood as a connective tissue. It is a connective tissue in a fluid form because it is consisting of the fluid as well as the liquid portion. It is called the fluid of life, fluid of health, because it is fighting against different diseases. When a foreign particle enters in the body, it is fighting against it. So coming to the blood, it is of red color, but it is colored red in the arterial portion because arteries contain the oxygenated blood. Moving to the volume of the blood, the normal adult contains the 5 liter of the blood, but in the newly born baby it is 450 ml. But with the passage of time, with the growth, it is converted into the 5 liter. The pH of the blood is 7.4. I have discussed it earlier in my previous lecture of the acid-base balance, that this is the normal pH of 7.4, which is maintained by the blood through the blood buffer system. Blood consists of 3 types of the cells. Number 1 here is the RBC, WBC, platelets. These are called the erythrocytes, these are called leukocytes, and the last one is platelet, which is called thrombocytes. Another portion of the blood is the plasma. Plasma is forming 55% of the total blood, while these RBCs are forming 45% of the blood. When the RBCs are expressed in a percentage form, this is called hematocrit. This is the hematocrit value of the normal hematocrit value of the human body. So these RBCs are forming the hematocrit, while the plasma consisting of the 55% portion of the blood. This 55% is formed from inorganic material, organic material, some liquid, gases like carbon dioxide, oxygen, nitrogen. All these are forming the plasma. Here is another component of the plasma, which is fibrinogen. This fibrinogen is differentiating this plasma from another fluid, that is serum. Plasma is actually a straw colored clear fluid. This plasma fluid and the serum fluid are same, but here is the difference, that is fibrinogen. This fibrinogen. Plasma contains the fibrinogen, while the serum do not contain any fibrinogen in it, because this serum is actually a fluid which oozes from the clotted blood. Now we will discuss the functions of the blood. Blood is performing different functions, but we are here with the major functions performed by the blood. This is the nutritive function, then respiratory function, then excretory function as well. The regulation of the acid-base balance, which is very important because I have discussed it earlier in my previous lecture, that it is regulating the acid-base balance by the blood buffer system. It is also transporting hormones and enzymes. Hormones, either these are released from the ductless glands, that is endocrine glands. The hormones are released and into the blood, and from the blood it is transported to different areas or in the peripheral parts of the body. Enzymes, lipase enzymes are also transported. This is the defensive function. This defensive function is very important, and it is usually performed by these cells, WBCs, monocytes, leukocytes. These are called the leukocytes. Neutrophils and monocytes are performing this function. This is actually engulfing the foreign particle like bacteria, viruses, etc. So this is the cell which is performing the defensive function in the blood. So these are all the functions performed by the blood. Here is the first type of the cell, which is red blood cells. Red blood cells are non-nucleated and they are called erythrocytes. These are carrying the oxygen from the alveoli to the different parts of the body through this hemoglobin molecule, which is a pigmented molecule, has affinity for the oxygen. So normal value of the RBCs is 4.5 to 5.5 million per cubic millimeter of the blood, and this value ranges from 4.5 to 5.5, but in the males it has 5 million per cubic millimeter value, while in the females it is usually low, which is 4.5 million per cubic millimeter of the blood. So these cells are like a biconcave shape. This is the usual shape of the RBCs, biconcave shape. This biconcavity is giving it significance that these can pass through the small capillaries without any damage. So it is also increasing its surface area and also allowing the oxygen to pass into these cells. So coming to the origin of these type of the cells, RBCs are originated from the bone marrow, and bone marrow contains the stem cells. These stem cells then convert into the different specific type of the cells. So this UPHSC stands for the uncommitted pluripotent hematopoietic stem cells. These are converted into the committed pluripotent hematopoietic stem cells. Once these are formed, they have some specific capability to convert it into the specific cell, like the clone-forming blastocytes. Clone-forming blastocytes are then converted into the different classes of the clone-forming units. Number one, here is the erythrocyte. Likewise, leukocytes and other type of the cells are formed. Here is the erythrocytes. Clone-forming unit of erythrocyte is giving the first stage of the immature RBCs. Immature RBCs are formed that are called the megaloblast. These pro-erythroblasts cells are also called megaloblasts. These contain 2 or more nuclei. HB synthesis starts from here. In the early normoblast, the 2 or more nuclei which are formed here disappears, and then the intermediate normoblasts are formed. The HB starts appearing in this phase of the cells. These intermediate normoblasts then converted into the late normoblasts. In late normoblast stage, the nucleus disappears and the reticulum is formed. Once the reticulum is formed, these are called the reticulocytes. The reticulocytes are the immature RBCs. So these reticulocytes are converted into the mature erythrocytes. These mature erythrocytes, once formed, it got the biconcave shape, and also there is no nucleus inside these mature cells, and it contains only HB for the transfer of the oxygen molecules from the alveoli to different parts of the cells. RBCs contains the special type of the cytoskeleton. The cytoskeleton consists of the 3 proteins. Number one here is the actin, and the second one is the spectrin. This actin and spectrin are anchoring to the transmembrane proteins through another protein which is called ankyrin. So here I will discuss here the problem that is hereditary spherocytosis. This is a condition in which there is the absence of this spectrin protein. Absence of this spectrin will result in the hereditary spherocytosis, in which the shape of the RBC is deformed, that is, this biconcavity is lost, and will result in the sphere shaped cells which are called spherocytes. These are globular cells. So this is the condition I have discussed here. So these are all the characteristics of the normal RBCs. RBCs have the lifespan of the 120 days. After that they will die and will be removed by the reticuloendothelial cells. The lifespan of the RBCs is 120 days, and after 120 days the cells become fragile, and the normal younger RBCs can pass through the capillaries easily without any damage. They can pass, but with the age, that is after 120 days, the cell become fragile, and while passing through these capillaries, these RBCs are broken down. So once the RBCs are broken down, the hemoglobin is released out of these cells. So this hemoglobin is further degraded, hemoglobin is released, and this hemoglobin is then further converted into the 3 parts, that is the iron, globin and porphyrin. This iron then combined with the protein, that is apoferritin, and this is converted into the ferritin, which is stored in the body and can be reused later. Likewise, this globin is also becoming the protein pool and can be reused later by the body. This porphyrin is degraded by the liver, and this is converted into the bilirubin and excreted through the liver. So normally the 10% of the RBCs are dying every day in the normal adult. So this percentage is giving about 0.9 to 1.5 milligram per deciliter of the bilirubin. So this is all the fate of the RBC, normal destruction of the RBCs in the human body. So coming to the functions of the RBCs, RBCs are performing the major function, which is Number one is the transport of the gases like oxygen and carbon dioxide. 97% of the oxygen is carried by the RBCs through the molecule or a pigment which is hemoglobin. Hemoglobin combines with the oxygen, they form the oxyhemoglobin. So 97% of the oxygen is transported to the different parts of the body through these cells, that is RBCs. Likewise, carbon dioxide is also transported in the blood through this hemoglobin. Once the carbon dioxide combine with the hemoglobin, they form the carboxyhemoglobin. Carboxyhemoglobin is excreted from the body. The next function is the blood group determination. Blood group is determined through these RBCs, because RBCs contain special antigens like antigen A, B, Rh factors. These are all factors allowing the determination of the blood group. These are all the functions which are performed by the RBCs. Next, we are going to discuss the polycythemia. Polycythemia is the condition in which there is an increased amount of the RBC count. The RBC count is increased. There are two types of the polycythemia, that is physiological polycythemia and the pathological polycythemia. Physiological polycythemia involves the factors like age factor, sex factor, like high altitude, increased muscular exercises, and environmental factors like high temperature and after the meal. So these are all the conditions which are increasing the amount of RBCs. So here we are going to discuss number one, that is the high altitude. In the high altitude, there is the hypoxia, and the people over there which are living in the high altitudes, they are actually having the value of 7 million per cubic millimeter of the blood, which is not the normal range of the RBCs. So at high altitudes there is a hypoxia and the stimulation of the kidney. While the kidneys are stimulated, and there will be the production of the erythropoietin. This erythropoietin is then resulting in the stimulation of the bone marrow, which will result in the increased amount of the RBCs production. In this way, the count of the RBC is increased in the high altitude. Like high altitudes in the emotional conditions and in the muscular exercises, strong muscular exercises, this will result in the increased sympathetic activity, which will further result in the secretion of the adrenaline. Adrenaline is secreted, and there will be the contraction of the spleen. Once the spleen is contracted, there will be the release of the RBCs, and we got the high amount of the RBCs in the body. RBCs are increased in these situations, and these are normal physiological conditions in which the RBCs are increased. These are not the pathological conditions. The second type of the polycythemia is the pathological polycythemia. In pathological polycythemia, there are primary polycythemia and secondary polycythemia. Primary polycythemia is also called the polycythemia vera, in which there is an increased RBCs about 10 million per cubic millimeter, in this situation there will be also increased WBC count in the body. So these are the pathological conditions we will discuss later in the pathological disorder of the blood. So these are normal physiological phenomena here, in which there is the increased RBC amount, that is called the polycythemia. Thank you very much for the listening, and we will discuss next the WBC.